Hello and welcome to today's webinar. I'd like to begin by giving a brief clinical trial update before turning it over to Alycone Therapeutics to share more about their device and their ongoing PIERRE trial. Next slide. So for today's webinar, first we will give a quick review of the FDA-approved drugs for SMA and how they work. Then we'll talk about where we want to go. So things like optimization of the current drugs and new drugs to meet unmet needs, or drugs that can be used in combination with SMA upregulating drugs to achieve a greater therapeutic effect. And then I will like to also introduce Alycone Therapeutics following the conclusion of those portions, who has a device in clinical trials intended to lessen the burden of repeated intrathecal injections for the administration of Spinraza or nusinersen. Next slide. So as I'm sure almost all of you in attendance know, the landscape of SMA has rapidly evolved in recent years. The FDA approval of 3 drugs has revolutionized and really changed the natural history of SMA. Yet we know that early identification and treatment really alter long-term outcomes, most strikingly in pre-symptomatic infants. But we also know that treatment of symptomatic individuals can provide improvements such as increased function, stabilization, or slowing of function loss. Yet there is still great unmet need, especially for individuals for whom treatment was not available prior to symptom onset. And we estimate that about 30% of all SMA patients remain untreated. Next slide. So you can see here our current SMA drug pipeline, where we capture all drugs from approval to preclinical stages. And you can see the 3 approved drugs here highlighted in blue, which are Spinraza, Zolgensma, and Evrysdi. These drugs all work by regulating or upregulating the levels of SMN protein. Next slide. So if we take a step back and we talk about SMA and the genetic cause of SMA and how these drugs work, you can see here we have the SMN1 gene and the SMN2 gene. And SMA is caused by loss of function of the SMN1 gene. So if you hit next slide, Sarah. So when a person loses their functionally loses both copies of the SMN1 gene, they're forced to rely on SMN2. And the key difference between SMN1 and SMN2 is a single nucleotide change, which results in an exon being left out of the transcript, which eventually forms the SMN protein. So you can think of it kind of like a missing puzzle piece. And so when that event happens, we call it a missplicing event. Essentially, the protein produced from SMN2 isn't as functional because it's missing that exon or that key puzzle piece. It's not bad or toxic. In fact, having more copies of the SMN2 gene generally results in less severe forms of SMA. However, it can't compensate for the loss of SMN1. And so a key therapeutic strategy and how the currently approved drugs work is by upregulating or increasing the levels of that fully functional SMN protein that are produced. Next slide. And so those are what we call SMN-dependent therapeutic strategies. And so they are things like gene therapy, replacing the missing SMN1 gene via viral vector, and that would be Zolgensma, or redirecting the splicing of SMN2 such that that missing piece, that missing exon or missing puzzle piece is included, and so the resulting protein is fully functional, and those are things like Spinraza and Evrysdi. Next slide. But then when we think about novel therapeutic strategies, we think about things that are SMN independent. So other targets, other ways to meet unmet needs of patients by targeting other things than upregulating SMN expression. So we think about other pieces of the of the equation that make up SMA. So things like neuroprotection, protecting the nerve against injury or degradation. We think about muscle and improving muscle strength and function. And then we think about the connection between the two, or what we call the neuromuscular junction, which is the point where the nerve connects and talks to the muscle. And then we think about regenerative targets, or ways to promote reconnection of nerve and muscle and promote muscle regeneration. And we know that it'll likely take a combination of these different kinds of approaches combined with our SMN enhancing approaches to achieve the best therapeutic outcomes for all individuals with SMA. Next slide. And so you can see that kind of summarized here in the chart, where we have our SMN dependent therapeutic strategies and our SMN independent therapeutic strategies, most of which we don't have any approved drugs for right now, but there are several ongoing clinical trials. And we know, like I just said, that it will likely take a combination of these to achieve greatest therapeutic effect. Next slide. And so here you can see highlighted the drugs in clinical trials. And these slides will be available afterwards as well if you want to take a closer look. Next slide. And so when you're thinking about clinical trials, and perhaps exploring the possibility of a clinical trial and whether that's right for you or your loved one, we do have a new resource that I want to highlight available. It's a clinical trial finder widget. It allows you to put in key things about yourself and what how far you're willing to travel, other things like that, what the types of trials you're interested in, and helps you locate clinical trials that you may be interested in participating in and gather more information about them. Next slide. And here's a list of further clinical trial resources that are available from PIERRE SMA. Everything about understanding inclusion and exclusion criteria in clinical trials, how we think about drug combinations, and other information from FDA about the drug approval process. And again, we will be sharing these slides so that you have access to those resources as well. And so now, next slide, Sarah. I'd like to introduce Alcyone Therapeutics, who is working on the development of ThecaFlex, which is a port catheter system that's designed to minimize the burden of repeat intrathecal injections. And they're going to provide an update on the development of that device, as well as their ongoing PIERRE clinical trial that's focused on evaluating the safety and performance of this device for administration of nusinersen. And so I'll kick it over to Chris Coletta with Alcyone for the next portion of the webinar.
Thank you, Jackie. Alcyone's honored to be a new partner to the Cure SMA community. We'd like to start today's webinar by thanking Cure SMA and to everyone in attendance today for providing us with the opportunity to discuss the ThecaFlex intrathecal port catheter system, which has been deliberately engineered from end to end with an aim to address the unmet need for an alternative to lumbar puncture for drugs requiring repeat intrathecal dosing. My name is Chris Coletta. I'm the Senior Vice President of Strategic Alliance here at Alcyone. I'll be joined today by my colleagues, Megan Holmes, who is the Director of Program Management, and John McGuire, who is the Senior Director of Clinical and Regulatory Affairs. We're going to begin today's webinar by providing you with an introduction to Alycone Therapeutics. Then we'll discuss unmet need of an alternative to lumbar puncture for repeat intrathecal dosing, with a specific focus on the SMA population. After this, we'll provide an overview of the ThecaFlex intrathecal port catheter delivery system that we believe has the potential to become an essential component of addressing that unmet need. Finally, we'll finish the webinar by providing an update on the ongoing PIERRE clinical trial that is a part of an Alcyone Biogen collaboration to evaluate the safety and performance of the ThecaFlex port catheter system for the delivery of nusinersen. Alcyone takes a solution-based approach to improving the lives of people with neurological conditions. We work at the interface of medical device and therapeutic technology to bring holistic solutions to areas of long-standing unmet medical need. So what does this mean? It means we start with a clearly identified unmet need that, if addressed, has the potential to drive a step change improvement to the patient experience and to the quality of care they receive. Then we deploy a multidisciplinary team to focus on the development of a targeted and effective custom design solution. Alcyone has a long history of focusing on these types of custom design solutions. Since we were founded in 2010, we've applied our solution-based approach to multiple areas. Starting the upper left-hand side of the slide, the Alcyone microcannula platform was designed to enable the targeted delivery of glioblastoma therapies directly to cancerous tumors located in the brain. And this has been deployed in the clinic to deliver investigative therapeutics. The reflow ventricular system provides a fundamentally new option for the management of hydrocephalus, which is a neurological disorder caused by abnormal buildup of cerebral spinal fluid in the ventricles within the brain. This technology was actually spun out into a separate company called Annuncia, and it offers the only non-invasive way to attempt to restore, increase, or maintain cerebral spinal fluid flow within a ventricular shunt system. Alcyone is also developing targeted delivery technology for intrathecally delivered therapeutics by combining our intrathecal catheter technology with a deep understanding of how the dynamic flow of cerebral spinal fluid impacts the transport of drugs within the central nervous system. Additionally, Alcyone is focused on the development of a gene therapy with the potential to address a therapeutic unmet need in a disease known as Rett syndrome, as well as a number of other X-linked disorders. Today, of course, we'll focus on the ThecaFlex port catheter system, which has been deliberately engineered from end to end as an alternative to lumbar puncture with the objective of minimizing patient burden related to repeat intrathecal injections. So we'll move on to the unmet need and to a brief introduction of the ThecaFlex port catheter delivery system. Currently, the only approved route of administration for nusinersen is via repeat lumbar punctures. And while lumbar puncture is the standard of care for the delivery of nusinersen, it can represent a barrier for some individuals. This is particularly true for those who may require extended time in a specialized hospital setting with anesthesia and/or radiation required for effective administration of the therapy. Lumbar puncture can also potentially be a barrier for those who experience post-lumbar puncture headache. Given the clear unmet need, the team at Alcyone undertook the development of the ThecaFlex system with the aim to provide such an alternative approach to standard lumbar puncture for repeat intrathecal delivery of therapeutics, and again, to potentially improve the experience for those receiving nusinersen for the treatment of SMA. In absence of an FDA-approved alternative to lumbar puncture, there have been over 70 instances reported in the literature of patients who have been implanted with off-label port catheter systems. The observation that these off-label systems have been used as a stopgap measure to provide an alternative to lumbar puncture helps to highlight the extent of the unmet need. Now, there are a couple of important things to note about these off-label systems. First, none of the systems described were specifically designed from end to end for repeat intrathecal injections. Second, these systems were not subjected to the rigorous non-clinical and clinical testing required to demonstrate that they are safe and effective for the purpose of repeat intrathecal injections. As part of an effort to establish an FDA-approved alternative to lumbar puncture for repeat intrathecal dosing, Alcyone and Biogen formally established a collaboration in late 2022 for the clinical development and commercialization of the ThecaFlex port catheter system. One of the main objectives of this collaboration is to ultimately gain FDA approval for an on-label alternative to lumbar puncture for the administration of nusinersen for the treatment of SMA. As will be detailed further a bit later in the webinar, there's an ongoing clinical trial that is evaluating the safety and performance of ThecaFlex. Jackie referenced it earlier in the presentation today. My colleague John McGuire will be providing an update on this trial, which is being referred to as the PIERRE trial. But for now, I'm going to turn it over to my other colleague, Megan Holmes, who will provide an overview of the ThecaFlex intrathecal port catheter system.
Thank you, Chris. As previously mentioned, the ThecaFlex system was deliberately engineered for repeat intrathecal injection and improve patient experience and convenience. The intent was not to provide a generic port and catheter, but rather look beyond and design a customized solution for SMA patients. A system that opens up options to those patients with and without complex spine. The ThecaFlex platform, shown on the left-hand side of the screen, consists of an implantable intrathecal catheter, fixation device, and port. The system itself was designed with extensive input from SMA experts to address the specific needs of the SMA population and to overcome some of the shortcomings observed in the use of off-label systems. The top image depicts an implanted system with the catheter in the intrathecal space, the fixation device secured beneath the skin near the catheter insertion site, and the remaining catheter length and port secured beneath the skin in the lower back. The port implant location can be selected based on each patient's specific anatomy and preferences. A loop of slack is intentionally left in the system between the fixation device and port, and this slack loop is there to provide additional catheter length as needed to accommodate patient growth and movement. The top right image provides an example of an infusion through an implanted ThecaFlex system. A specific needle designed such that it will not damage the port will be inserted through the skin and into the port, and then the nusinersen infusion will be conducted per the drug label. On this slide, we'll take a closer look at some of the specific design features for the implanted system. The catheter was designed for consistent aspiration and infusion using the outlet hole configuration shown here. There are 12 outlet holes at the tip of the catheter that are primarily intended to allow for the tip to flex and safely track into the intrathecal space without damaging the surrounding anatomy. There are an additional 18 outlet holes placed in a helical pattern around the catheter surface. If the catheter is positioned against tissue, this ensures that some outlet holes remain available to aspirate cerebrospinal fluid, or CSF, and infuse drug through. Additionally, the materials and construction used in the catheter were designed to allow implanters to place the catheter regardless of the complexity of the spine and to maintain its integrity after being implanted. The port was designed to ensure the ability to make and maintain a secure connection between the port and catheter. Additionally, the shape and size were designed to allow the infusionist to be able to readily identify the center of the port septum for infusions, but also to minimize the intrusiveness of the port for patient comfort. And then the fixation device, shown on the lower right-hand side of your screen, was custom designed to hold the catheter with enough force to prevent it from moving once placed, but also to not damage the catheter or restrict flow. The implantation procedure will be familiar to most implanting surgeons. Vascular ports and catheters, as well as intrathecal catheters, have been around for a long time, and the procedures for implanting them are well practiced with a history of safety. The surgery will be performed under anesthesia, and with the patient under anesthesia, the surgeon will insert the catheter into the intrathecal space where the CSF and spinal cord are located. Once the catheter is in position, the fixation device will be used to secure the catheter fully, and then the surgeon will track the free end of the catheter underneath the skin to the port pocket. The catheter will be connected to the port, leaving slack in the system to allow for patient growth and movement. Then the incisions for both the port pocket and fixation device pocket will be closed, and the implanted system will be fully beneath the skin. As noted previously, the implant location for the port can be customized for specific patient needs and clinician recommendations. The image shown on this slide is one potential implant location in the lower back. Based on experience from patients in the PIERRE clinical trial to date, the expected duration of the implantation procedure is between 1 and 2 hours, with discharge within 24 hours for most patients thus far. Now, nusinersen infusions through the ThecaFlex system are expected to take place in an exam room setting. The healthcare provider accessing the system will clean the skin around the port and may apply a topical anesthetic to the skin. They will then locate the port with their fingers, feeling for the titanium ring feature to identify the center of the port, and inserting an infusion needle through the skin and into the port. Drug infusion will follow the standard nusinersen labeling, and the needle will then be removed from the port, and a temporary bandage, typically a Band-Aid, will be placed over the needle site if needed. Based on experience from the patients implanted in the study to date, the entire procedure is expected to take under 30 minutes with an injection time of between 3 and 10 minutes. Since this isn't a lumbar puncture procedure, you're not likely to be required to lay down for an hour post-infusion. Now I'll hand over to John McGuire to discuss the ongoing clinical trial of the ThecaFlex system.
Thank you so much, Meg. Good afternoon. I'm going to talk about the PIERRE clinical trial to assess the safety and performance of the ThecaFlex DRX system. As mentioned, the PIERRE clinical trial is underway. The objective of this study is to evaluate the safety and performance of the ThecaFlex system and generate the data that the FDA will use to determine if it is suitable to advance the technology to market. We have completed the initial stage of the study. The FDA is currently reviewing the data from the initial stage before providing a decision on approval to continue enrollment. The data has been reviewed by an independent data monitoring committee, and they have provided a recommendation to continue the study without changes. The success metrics for the PIERRE trial is fairly straightforward. It will be determined by the proportion of patients who are able to be successfully implanted with the ThecaFlex system and then are able to receive successful infusions of nusinersen through 12 months. This is typically about 3 infusions at a minimum. We will also be looking to see how much anesthesia and/or radiation is reduced with the ThecaFlex system when compared to lumbar puncture. If the data from the PIERRE study supports market approval, we are anticipating being able to launch in 2027 and address the unmet need a little bit more broadly. If the system is intended to be lifelong and therapy infusions will continue through the system post-study completion, your treating clinician will make the final determination and can determine if the ThecaFlex system is in the patient or your best interest. As mentioned earlier, the ThecaFlex was developed with extensive input from SMA experts to address the specific needs of the SMA population. Before advancing the ThecaFlex system to clinic, it underwent extensive testing to clear the justifiably high bar set by the FDA prior to starting human clinical trials.
For this testing, Alcyone followed existing standards and worked with clinicians to fully understand how the device would be used and all environments and forces it would be exposed to. For bench testing, all components of the system were tested for strength, functionality, and the ability to withstand forces and manipulations they will experience during the implantation and infusion procedures, as well as within the body as a permanent implant. Biocompatibility testing was conducted per well-established standards to verify that the materials in the system are safe to implant within the body. Drug and fluid compatibility testing was conducted to ensure that the device is not damaged by and does not cause damage to the drug or any fluids that pass through the system. Additional testing, including MRI compatibility, mechanical and material analyses, and many others were completed by Alcyone and reviewed and approved by the FDA prior to initiation of the PIERRE clinical trial.
Thank you, Meg. The findings from the completed Part A of the PIERRE study showed the device appeared performing as intended. All implantation attempts were successful and are functioning according to intended use. There have been no device-related adverse events that have been reported to date. After review of this data, the independent data monitoring committee recommended continuation to Part B without any changes. The FDA is currently reviewing the data and the DMC recommendation and reviewing and determining approvability to continue to Part B. If the FDA approves, we anticipate being able to continue enrollment at currently activated sites starting in the summer. We will continue to activate additional sites starting in the early summer, targeting center activations in the states that are highlighted in the orange on the map shown on your screen. We recommend you check clinicaltrials.gov for an up-to-date list of activated centers. If you are interested in participating, keep an eye on clinicaltrials.gov for an updated list of centers. We will continually update this list as centers become activated. If you are at a center, if the center where you receive care becomes active, reach out to your care team to see if you're a good candidate for the study. If so, schedule some time with your care team to learn more about the study and discuss your potential participation. For patients who enroll in the PIERRE study, other than the implantation procedure and potentially port access, the first port access, the PIERRE study should not require any additional visits compared to the existing standard of care. Here we have the QR code. You may scan this QR code with your mobile devices for more information about the PIERRE trial. The QR code will take you to the PIERRE study site on clinicaltrials.gov. As mentioned, we will regularly update this site to provide the current listing of all sites that have been activated for the study, including contact information.
Thank you, John. Before we summarize what we've covered in today's webinar, I'd like to highlight a few of the future Cure SMA events that we're looking forward to participating in throughout the year. In the near term, we'll have a team at the upcoming annual Cure SMA conference in Austin, Texas from June 6th to June 8th. Also, just before the conference, Alcyone representatives will be in attendance at the Cure SMA Walk and Roll event in Villanova, Pennsylvania on June 2nd. Later in the year, we'll be presenting at a few of the Summit of Strength events, one in Arizona and a couple in California, one in Northern California and one in Southern California. All right, so this brings us to the end of today's webinar presentation. Over the course of the discussion today, we hope that we've provided everyone with a better understanding of the collaboration between Alcyone and Biogen that has the aim of addressing the unmet need for an alternative to lumbar puncture. Through the development of the ThecaFlex intrathecal port and catheter system, which has been deliberately engineered from end to potentially improve the experience for those receiving nusinersen for the treatment of SMA. With Part A of the PIERRE clinical trial completed, we're eagerly anticipating the start of Part B sometime in the summer of 2024. Specific and up-to-date information with respect to the PIERRE clinical trial can be found at clinicaltrials.gov.